Good morning, grade 12 students. In this session, we're going to continue with our chapter about the immunology, and we move to chapter 7, activity 1, the non-specific immune response. Let's make a quick revision about the activities 1 and 2. Activity 1, we said that the immune response is divided into two different types, in specific or non-specific. Non-specific, as its name indicates, it doesn't depend on the identity of the intruder. Effector cells, they are the monocytes and the granulocytes. What are the reactions that occur? We have inflammation and we have the phagocytosis, eating other cells. We said the non-specific, it's local. However, the specific immune response depends on the identity. Effector cells, they are the B cells and the T cells, and it's systemic. In about the specific immune response is divided into humoral or cell-mediated. The humoral is carried out by B lymphocytes by the secretion of antibodies inside the serum. So we have antibodies. However, the non-specific the cell-mediated, immune response is carried out by TC cells. TC cells in the T cytotoxic cells, Ahmad in the car, for example. So carried out by T cytotoxic cells. TC cells, they kill infected cells by the secretion of perforin and granzymes. Perforin, it creates the pores inside the cell, and granzymes, it go to the degradation and the killing of the cell. Here about the natural barriers, here about the phagocytosis, its steps and the differences and why some types of bacteria we are, for example, not killed by phagocytosis, maybe because there is enzymatic deficiency or we have a resistant bacteria. Document 2, we talk about the specific immune response. In this experiment, we have the killed salmonella, it protects against salmonella, while the killed Vibrio cholera, it protects only against the Vibrio cholera. While if we inject killed salmonella and then 10 days later we inject live Vibrio cholera, this will lead to the death. Why? Because the killed salmonella, it provides immunity only against the salmonella and not against the Vibrio cholera. And this shows that the immune response is specific. In this experiment, we said that we have serum and we divide the blood into serum and the blood and the cells. And whenever we inject the cells and injecting with tetanus, it died. However, injecting the serum and then injecting with tetanus toxin, it survived. We said that the serum protects. Serum, it contains antibodies. So antibodies mean that we have B lymphocytes. So it's a humoral, specific humoral immune response. However, in the CGB and cog bacillus, we said that they share the same epitope, and injecting the cells, it provides immunity. However, injecting the serum, it leads to the death of the animal after the injection of KB. We said that only the cells that protect, and to be more specific, they are the T cells. So it's a cell-mediated immune response. We said the meaning of toxoid or anatoxin or attenuated, meaning that this toxin is very weak. It's like the case of vaccine. It protects, it provides immunity against a specific type of cells. So the big question here, what determines which type of the immune response it will be? If it's a specific humoral immune response or a specific cell-mediated? What determines which type of the immune response is going to be conducted here? Let's move to document 3 to talk about the induction of the specific immune response. Induction in the specific immune response. Document 3 said that the Mosier experiment. Mosier and the experiment, Mosier and his collaborators were the first to demonstrate in 1967 the important role of the macrophages. Macrophages that do the process of phagocytosis in the induction of the specific immune response. Their experimental system involved the production of mouse antibodies against the sheep red blood cells. Sheep red blood cells, which is the non-self with respect to the mouse. SRBC, sheep red blood cells. Mosier experiments and the results, they are schematized in document 8. Here we remove the spleen of this mouse. The spleen was removed. We have cell suspension containing macrophages and lymphocytes. We divided them into three different culture mediums. The first culture, it contains only macrophages, and we culture it in the presence of sheep red blood cells for 4 days. So here we have macrophages, only macrophages. and we cultured them in the presence of sheep red blood cells, so we have no antibodies. However, when we culture the lymphocytes, and to be more specific, they are the B lymphocytes, in the culture medium in the presence of sheep red blood cells for 4 days, we have the production of antibodies. So we have antibodies. But these antibodies, no antibodies here, we have to correct. We have small amount of antibodies, not no antibodies at all. It should be small amount of antibodies. Okay? So macrophages alone, there is no antibodies. However, in the lymphocytes, we have not no antibodies. We have to correct it here. Small amount of antibodies. So what is the responsible for the secretion of the antibodies? They are not the macrophages, but are the B lymphocytes, to be more specific. However, in the second culture medium, this is the second culture medium. In this culture medium, what do we have? In this culture medium, we have macrophages with lymphocytes. So we have anti-sheep red blood cells antibodies. So we have a high amount of the anti-sheep red blood cells antibodies. So this can be explained that antibodies, they are secreted by lymphocytes. This is true, but they are secreted in a high amount in the presence of macrophages. So macrophages, they induce or they provoke or they help or they induce or they facilitate the secretion of antibodies in a high amount. This is the importance of the macrophages. So here I did in part one describe the probing the documents, and as well as I did the interpret. So description: from a mouse, we took a spleen, and we have cell suspension containing macrophages and lymphocytes is obtained from the spleen. And the suspension separated into three containers. The first contained only macrophages, the first culture medium. The second contained macrophages and lymphocytes, while the third contains only lymphocytes. The cells in the three containers, they are cultured in the presence of sheep red blood cells. And the first container contains no antibodies. The second container contains a high amount of antibody, while the third, they said no. I said you have to correct it, low amount of antibodies. This is described. Interpret, analyze, classification. In the container that contains only macrophages with sheep red blood cells, there is no antibodies. However, at the same time, in the container that contains only lymphocytes with sheep red blood cells, we have low amount. So this indicates that the lymphocytes are not the macrophages. They are the cells that secrete antibodies. However, in the second container that contains both macrophages and lymphocytes, we have high amount. This indicates that the production of high amount of antibodies, it needs the presence of macrophages. Since the macrophages, they activate, for example, the lymphocytes to secrete high amount of antibodies. Remember, when we talk about the interleukins, interleukin 4, interleukin 2. So we're going to talk about interleukins in specifics. So macrophages role in the induction. When a macrophage phagocytoses, meaning that eats other cells, and digest a cell or protein, resulting peptides, they are attached to HLA class. We have class 1 and class 2 molecules, and they are presented on the cell surface. The macrophage will migrate to the lymph node where it becomes an APC, antibody antigen presenting cells. We said that it's APC. APC, antigen presenting cell. T helper, TH, they circulate between the lymph nodes where they inspect the HLA peptide of the APC. Only T helpers that are specific for this presented by the APC, they remain intact and then they are activated and they're proliferated. Pay attention to the T helper. T helper, they will have an important role. T helper, they are the one which determine whether the type of the immune response is going to be specific humoral or specific cell mediated. Fate of the activated T helper lymphocytes. Activated T helper cells, TH. We said that we have TC, T cytotoxic, TH T helper. they proliferate, يعني meaning that they multiply, and each one of them will have a cellular clone made of a large number of daughter cells. All of them, they are identical to the mother cell. This activation of the antigen-specific lymphocytes is called بنسميها clonal selection. Clonal selection, some cells become memory cells. هيدي البروسيس whenever they multiply and they are activated by specific antigen بنسميها clonal selection. This يعني meaning that we have selection and this selection is a clonal. Clonal meaning that we have copies of the cells and these copies of the cell they are specific to the mother cells. Some of these cells, whenever they are multiplying or they are cloned, either they divide into memory cells or they are differentiated into interleukin secreted cells. Here now about the interleukins, and we're going to look about this in details. Others, they complete differentiation and they secrete the cytokines. حكينا عنها before قلنا هنحكي عنها in the coming sessions. Cytokines بنسميهم interleukins and they play an important role in the immune system. Interleukin secreting cells, they only survive for specific number of days, 3, 4, 7, 10 days. These interleukins, they are presented like this. Interleukin, they give it as IL. Interleukin. Interleukin, we have interleukin 4 and we have IL interleukin 2. We're going to talk about what is the function of interleukin 4 and what is the function of interleukin 2. These are secreted by the T helper. As I said before, T helper, they have an important role in determining what is the type of the immune response, whether it is a humoral or a cell-mediated immune response. So I need part one already we interpret and I added for the interpretation, I did it describe and I made describe my interpret. I will move to part four. Formulate a hypothesis explaining the role of interleukins and the memory cells. What do they do? Interleukins, as I said, we have interleukin 4 and interleukin 2. Here, this suppose already I know the answer and it's correct. And you have to study these two. Interleukin 4, it has an important role in activation of B lymphocytes in the plasma cell. So, whenever the T helper secretes interleukin 4, this will activate the B lymphocytes into plasma cells. So, what do we have? We have antibodies. So, what is the type of the immune response? Is specific humoral immune response. However, when the T lymphocytes T helper, when the TH T helper, they secrete interleukin 2. This interleukin 2 will activate TC cells, T cytotoxic lymphocytes, into killer cells. So the TC cells, they are the active cells. So the type of the immune response here is specific cell-mediated immune response. So pay attention here. Whenever we have interleukin 4, activation of B lymphocytes into the plasma cells, into antibody secreting cells. So this is the specific humoral. While when the T helper they secrete interleukin 2, so T cytotoxic T becomes a killer cell and will kill other cells by the specific cell mediated immune response. So wait a minute. Interleukin 2, pay attention. 2, it starts with the letter T. So letter 2, it starts with the letter T. So 2 for the T. So whenever we have interleukin 2, we have activation of T cytotoxic cells, TC cells into killer cells. So it's specific cell mediated. While when we have interleukin 4, so B lymphocytes into plasma cells for a specific humoral immune response. That's why I said the T helpers they have an important role in determining whether the immune response is a specific humoral or a specific cell mediated. However, the memory cells they become effector cells one after the second contact. هيدا اللي سميناه secondary immune response. If you remember primary immune response and the secondary immune response. Part 5, what do you want? Do you want to, based on the information provided, name the different phases in the evolution of the activated T helper? T helper, what do they undergo? First, recognition. They recognize the antigen presented on HLA class 2 by a process شو بنسميها double recognition. Already we talk about this. Then we have the clonal selection. The clonal selection, we have clones, only these cells whose TCR is specific. What is the TCR? T cell receptor. It is specific for the antigen. They are activated and proliferated. That's why it's called clonal selection. They select only the T lymphocytes where the TCR is specific for the antigen, and they proliferate and they multiply. While differentiation, the cell that is active is proliferate and might undergo mitosis, multiply, and they differentiate into either effector cells or into memory cells. Document 4, role of the T helper in the immune response. Role of the T helper. Role of T helper in the specific immune response. T helper, pay attention to this statement. They are the orchestra conductors who are in charge of regulating the specific immune response. Activation of such cells allow the secretion of interleukins. هيدي اللي انا حكيتها few minutes ago. They are the orchestra conductors. انتوا بتعرفوا الاوركسترا كوندكتور يعني الموسيقار بيكون واقف على حد الاوركسترا هو اللي بحدد لهم what to do to increase the beat or to lower the beat to faster or to for example to accelerate or decelerate the speed and so on. So T helpers, they are the one which determine the specific immune response, whether it's a humoral or cell mediated. So importance of the T helper cells in the specific immune response. Nude mice, they are laboratory mice, right? اللي nude mice يعني nude mice يعني mice مش لابسين. Doesn't mean like this. They are nude mice, they have a congenital defect resulting in the absence of the thymus. ما عندهم thymus. يعني ما عندهم thymus يعني they lack the T cells. As might be expected, these might have a figure, by the way, in used mice that is susceptible to infection يعني عنده كتير infection. هذا هو example about the new mice. يخلقوا ما عندهم thymus يعني they lack the T lymphocytes. So these mice they don't have specific cell mediated immune response. ما عندهم cell mediated لأنه ما عندهم T lymphocytes because of the absence of T cytotoxic cells. But also, their specific human immune response is approximately null despite the presence of normal T lymphocytes. Why do you think this? ليش ما عندهم كمان humoral immune response? Why do you think this? We talked about this a few minutes ago. Why do you think that they also don't have a humoral immune response? Because we said that the T helper, they are the orchestral conductors. They secrete interleukin 2 and interleukin 4. Interleukin 4, it leads to the activation of the B lymphocyte to secrete antibodies and to become plasma cells. So it becomes a humoral immune response. So they don't have T lymphocytes. So the injection of tetanus toxoid. Toxoid is the attenuated, it's the anatoxin, is the neutralized one, is the non-active one, it's the weak toxin. It induces a specific humoral immune response. And that of CGB اللي حكينا عنه هو الKB carbacelis, they induce a specific cell-mediated immune response. In order to understand the role of the T helper cells in the determination of the type of specific immune response, the following experiments they are performed. T helper lymphocytes they are cultured in the presence of antigen and macrophages. حطوهم in the presence of antigen and macrophages. Few days later, the culture medium contains interleukins secreted by the activated T helper cells. The culture medium is added to a mixture of T cells and B cells. Each cell population is counted 40 hours later. Its proliferation indicates that this population is activated. Look to this figure in document B, experiment illustrating the role of T helper cell. حطينا first tube, we put the killed CGB وقال السي جي بي شو بيعمل cell mediated immune response. Then culture medium containing a high level of the interleukin 2. So we have culture medium containing interleukin 2. We transfer the supernated into another tube. So we have T cytotoxic cells and B cells. At the end, we have proliferation of T cytotoxic. So interleukin 2 proliferate the T cells and TC cells, the T cytotoxic. However, in the tetanus toxide that is needed for the humoral immune response, we put a culture medium containing high levels of interleukin 4. فحطيناه مع interleukin 4. We transfer the supernatant. لأنه هو supernatant في البالت في السوبرنيتد هو وجه التيوب or وجه الكالتشر أو وجه الكونتينر. But then we have B cells and DC cells. We have only proliferation of the B cells. So من هون بنعرف interleukin 2 it induces the proliferation of TC cells. So 2 interleukin 2 T يعني ببلش بحرف التي T cytotoxic cells. interleukin 4 B cells. So من هون نحن we get the hypothesis اللي اعطيتكم اياها انا بقبل الدوكيومنت. Okay? So هون لحتى نحكيهم. First of all, they said one-newt mice can only live in a sterile environment, an environment that is sterile, for a sterilized one, for example, in order to prevent any inflammation or any disease or any infection. Since these human mice, they lack B or T lymphocyte, ما عندهم لا humoral ولا cell mediated. Write a paragraph describing the experiment illustrated in document 8. So our first question, the nude mice, they don't have either humoral or cell mediated. بيجي حدا بيقول لك استاذ ما في قلت لنا نحن النيود مايس ما عندهم تي lymphocytes. Yes, they don't T helper lymphocytes. T helper ان هي الاوركسترا هي بتعمل activation للبي lymphocytes or للتي lymphocytes. تعمل activation للبي lymphocytes they become plasma cell. ما عندي T helper ما في activation لأنه T helper they secrete the interleukin 2 for the T interleukin 4 for the B. ما عندي T helper ما في لا B ولا T lymphocytes matured or activated. Description: two culture media containing T helper and macrophages are taken. دايما describe should be in the passive voice. And we don't have we cultured, we removed, we separated. No, you didn't. They did. I don't know who. So always in the passive. Killed CGB are added. So the second culture medium tetanus toxide is added. In the medium that's contained killed CGB IL2 interleukin 2 is obtained. فطلع عندي interleukin 2 while the medium that contains tetanus toxide طلع عندي interleukin 4. To other culture media containing TC and B lymphocyte, the supernatant containing IL2 is added to one of these culture media where the proliferation or the multiplication of TC is observed. The supernatant containing interleukin 4 is added to other culture media where we have only proliferation of B C cells. هعملها interpret. Okay? So the medium containing interleukin 2 TC cells proliferate at the same time, and the medium contains interleukin 4 only B cells they proliferate. So this indicates that interleukin 2 it provokes or stimulates or enhances the proliferation of TC cells, T cytotoxic, but not B cells. However, interleukin 4 هيدي لازم احفظها. Interleukin 4 provokes the proliferation of B cells, sorry, not T C cells. So B lymphocytes and not the T lymphocytes. So من هلا ورايح, whenever we talk about interleukin 2 and 4, they are secreted by T helper. Interleukin 2, it activates or provokes the proliferation of TC cells. So دايما انت حطها براسك عشان ما تضيع فيها. Interleukin 2, so for TC, T, 2T. Interleukin 4, B lymphocytes. So conclusion, T helper, it determines the type of the specific immunity. As a humoral, by secreting interleukin 4, so this will activate the B cells, or cell mediated by secreting interleukin 2 and activation of TC cells. 43. What do they need? They need, referring to document B, explain why nude mice don't have a humoral immunity. We said that nude mice, they lack T helper. So lack T helper, so they lack the T4, interleukin 4. Interleukin 4, it will enhance the proliferation or the activation of B lymphocytes into plasma cells, and thus will prevent the secretion of antibodies, which is the humoral immune response. Moving now to paragraph 2, fate of the activated B and TC cells. Among billions of cells expressing different membrane Ig immunoglobulins, Only those that are specific for the antigen, they are undergoing clonal selection and they are activated. A selected B cell proliferate in response to interleukin 4 وحكيناها before. And some cells of the resulting clone, they keep their membrane immunoglobulin and they become memory B cells. Other, they complete differentiation into short-lived plasma cells that secrete antibodies that are specific for the antigen. A plasma cell secretes about 3000 antibody molecule per second يعني تعطيني 3000 antibody بالثانية. Among billions of TC cells that recognizing this different TCR or expressing different TCR, only those that are specific for the antigen, they undergo clonal selection. Selected T cells, they proliferate one when the interleukin 2 is secreted by the T helper. Some of them become memory cells, and other they become differentiated into T cytotoxic cells or effector T cells that kill other cells and they secrete the peripheral enzymes and kill the other cells. So idol figure is showing the evolution of the activated B C cells in document C. So B C cells عملوا proliferation, عملوا multiplication. Some of them they are memory cells and other they undergo differentiation into effector B cells into plasma cells that secrete antibodies. While the antigen presenting cells, APCs, they undergo multiplication or proliferation into either memory cells or activated T C cells. This CPA also via APC بس كاتبينها هن بالفرنش. So activated T C cells or the memory cells. They said pick up the common traits and the main difference between the activation of T helper, T cytotoxic, and the B cells. So let's move to do this. Common between them, all of them, they have clonal selection. So they have selection according to the recognition of the antigen. And all of them, they have the same activation step. Differences, pay attention to this. B cells, they recognize soluble or cellular antigen. Soluble, it's free. Cellular, it's bound to the cell. However, T cytotoxic or T helpers, they recognize antigen presented on HLA class 1 or class 2 as respectively by the APC antigen presenting cells. TC is activated to interleukin 2, while B cells deactivated in interleukin 4. T helper secretes either interleukin 2 or interleukin 4, and this will determine whether the type of the immune response is specific humoral or specific cell mediated. So you have to know that intracellular microbe or antigen, viruses or some bacteria, they activate the cell mediated. However, bacteria and fungi except like an extracellular microbe, they activate the humoral immunity. This is a general information. You don't have to study it, but you have to know that the T helper they secrete either interleukin 2 or interleukin 4. This interleukin 2 activate T cytotoxic and will build the cell mediated. Interleukin 4 activates the B lymphocytes into plasma cell to secrete antibodies. Document 5, the specific humoral immune response. Already we talked about this a lot, the specific immune response. We have antibodies, they are secreted by plasma cells. They go to the lymph, the blood, and other fluids. How do the antibodies interfere in the elimination of the antigen? Neutralization by specific antibodies. Neutralization meaning that the binding of the antibody to the antigen. A variety of toxins that threaten the human health can be found. This document 5 and 6, they are in general. So we're going to move them quickly, documents 5 and 6. So these toxins are for as toxin of the snake venom, those of poisonous mushrooms and microbial toxins, antibodies are very efficient against circulating toxins. Toxins that enter the body, attack the target cell through the membrane receptors and alter their function. You have to know that antibodies they are specific for a given toxin, they bind to it and cover the attachment site, thus it prevents its action. They are said to neutralize. Neutralize meaning that to stop the action of the toxin. Also, antibodies are very efficient in preventing of the intracellular pathogens. The intracellular bacteria and viruses, they penetrate the target cell and the binding sites of the surface of the specific receptor expressed by the membrane of the target cell. Whenever they bind, they prevent it and they neutralize its function. Elimination of the intruder by the binding to this cascade. And document 6, I want you to know the specific cell-mediated immune response. Specific cell-mediated immune response is by the T lymphocytes. We said that the T lymphocyte, they have specific receptors, they call the TCR. They have granules. They contain toxic substances. They are called cytotoxin. These cytotoxins able to kill the infected cells. They are perforin and granzymes. Already صرنا حكينهم كتير. مرقت لكم اليوم بالintroduction also مرقتهم قبل. These TC cells, they recognize an infected body cell and bind to HLA class 1 complex through the TCR. It releases the perforin. شفنا البرفورين is released into the cell. Perforin will create pores in the cell. Then the released granzymes will enter inside the cell, will degrade it, and will cause the death of this cell. So this is the role of the granzymes. They penetrate into the target cell through the polypeptide or the perforin the channels. They trigger enzymatic chain reaction leading to the degradation of the DNA. This will cause the cell death. TC cells detaches from the killed target cell and recirculate again. It's again ready to kill any infected target cell carrying the same HLA class 1 peptide. Cancer and the immunity. This is the last idea I want to talk about it. The immune response not only recognizes elements that come from outside the body, referred to as non-self, but also abnormal elements emerging inside the body. These are called the modified self. إذا تتذكروا whenever we talk about the self and non-self مرة لكم شيء اسمه modified self. And this is example about the cancer. Cancer cells, from where did they result? Cancer cells, they result from intensive and uncontrolled proliferation. يعني mitosis عم بتصير كتير بسرعة. This one mistake will lead to million of mistakes. بغير لي بالsequence of the nucleotides. So تغير كل السيكونس حولت من self to a modified self, production of abnormal proteins, and this peptide deriving from modified proteins, they are arranged to MHC class 1 molecules and presented into the cells. These are recognized as modified cells. So cancer ما في يعتبروا أنا non self هو modified self, and they are killed by the TC lymphocytes. So it's very difficult for the body to eliminate the cancer cells because they, well, always said they have an enormous, rapidly development or proliferation, and they overwhelm the immune response, the immune system as well. So classical cancer treatments include either surgery or the radiotherapy or the chemotherapy. These procedures under serious side effects. So here عم يشتغلوا على شيء هي it's talking about in cancer immunotherapy one of the most promising approaches هي عم يشتغلوا على شيء خاصه بالstem cells. So what do they do? First, a sample of the tumor is removed. It contains cancer cells. هيدي بالفigure cancer immunotherapy. So cancer cell and it's initiated cancer specific T lymphocytes. Then the cell medium is cultured in the presence of interleukin 2. So هون أخذوا السل عملوا لهم culture in the presence of interleukin 2. كلنا بنعرف نحن الانترلeukin 2 it activated the T lymphocytes. So T cells they are activated and proliferate. They will kill the cancer cell in the culture. So إذا قدرنا نعمل هيدي البروسيس هلا بقول فينا نعملها هلا هن they are working on to take a sample, put it inside the culture medium in the presence of interleukin 2. Interleukin 2 هيتعرف على هيدا الكانسر cell and is going to kill it. After a month, the culture contain activated TC cells. بس هيكون عنده TC cells. يعني all the cancer cells or the tumor is removed. So the sample of the tumor is removed. It contains cancer cells. روح لي كل الكانسر cells. So these activated TC cells, they are harvested and then we inject them into the patient. They will attack the cancer cells and kill them since they recognize these as a non-self اتعرفوا عليهم and they will kill it. This process is easy. نحن عم نحكي حكي. It's easy بنحكيها, but it's hard to be applied. They are working on this process. So till today, we finished documents 3, 4, 5, and 6. 5 and 6 I moved quickly over since we finished all of them before and we moved only to talk about the figures. Tomorrow In the next session, we're going to move to document 7 to talk about the immunological memory. Even though we covered this, immunological memory, we talk about the primary and the secondary immune response. Then we're going to move to the exercises of the chapter.